start today by looking at the special groups in some amino acids. There are some amino acids that contain some special group, and that is where we are going to start today. So first of all, we have the arginine, and it has the special group guanidinium group. The phenylalanine has benzene as a special group. Tyrosine has phenol as a special group. Something like tryptophan, has an indole group. Histidine has an imidazole group, while proline has the pyrrolidine group. Now we're going to look at this group one after the other. Now look at arginine. Arginine contains the group which we call the guanidinium group, which is this. It is actually positively charged group. Okay. Now you have the phenylalanine. The phenylalanine has the benzene group as a special group attached to it. Now we have the tyrosine. Tyrosine has a phenol group attached to it. Tryptophan has the indole group, and the indole group is this. That is the indole group that is attached to tryptophan. Now, for histidine has the imidazole group. Imidazole group, and that is this group that is attached to it. While proline has the prolilidine group that is attached to it. So let's see the structure of the pyrrolidine group, which is that is the structure of the pyrrolidine group. So now let's look at the properties of amino acids. Generally, properties of amino acids. Now, the properties of amino acid, we have the physical properties. They are colorless, crystalline. Some of them might be sweet, like glycine, alanine, valine. Some are tasteless, like the leucine, and some are bitter, like the arginine and isoleucine. Now you have the aspartame, which is a sweetener that you find in most additives that you have in food. Okay, and that contains aspartic acid and phenylalanine. Even the monosodium glutamine, which you see in the white Maggi, the Maggi that they call white, the Ajinomoto, is also an amino acid, a sort of an amino acid, glutamine. Okay, so those ones are used in sweetening. Now, some are water soluble, while some others might not be water soluble. So they can be soluble in water, acid, alkaline, but insoluble in organic solvents. Okay, not all, but some of them. Now they also have very high melting point, as high as 200 degrees centigrade. Now let's look at isoelectric point. Now, amino acid exists as an ampholyte or a zwitteron in solution depending on the pH of the medium. What does it mean to be an ampholyte, or what does it mean to be a zwitterion? Now, the zwitterion means that it's not having any net charge, or not having any net charge could mean that the number of positive charges is equal to the number of negative charges. Okay, so we say the pH at which an amino acid exists as deuteron with no net charge on them is called isoelectric pH. Isoelectric pH, or it is known as this isoelectric point. So at the isoelectric point, the amino acid does not have any net charge. So we call them at that point, we say that they are zwitterions. So zwitterion, like I said, they have no net charge. No net charge. No net charge. It means the number of positive charges is equal to the number of negative charges. Now, in acidic medium, the amino acids exist as cations, and in alkaline medium, they exist as anions. So let's take a look at this example. When an amino acid is in the cationic form, cationic form means that is in a situation where it has a positive charge, and this positive charge is donated, okay, to maybe the solution. And when that happens, you see you're removing hydrogen ion. It will make this positively charged amino acid to possess just one negative charge and one positive charge, and there is no other positive charge in the solution. So at this point, pH 7, it is neutral, and we say it is in the zwitterion form. Okay, or you can call it an ampholyte at this point. Now, if you continue to remove the negative, if you continue to donate positive ion in form of removing hydrogen ion, you will find out that the amino acid will then exist as a negative, carrying negative charge. Okay, so, and that is the anionic form. So as the how will I say, as the pH continues to increase, do you understand? As the pH continues to increase, the amount of negative charges will also continue to increase. So you can see from the cationic form, when it is carrying one positive ion and it donates that positive ion in the form of hydrogen ion, it will get to the neutral state. From the neutral state, it will get to the anionic form. But when we talk about isoelectric pH or point of an amino acid, we said it is the point where there is no net charge. It is the point where there is no net charge, or where the net charge is 0. Okay. Now we say titration of amino acid. Say if hydrochloric acid is dropped wise on amino acid solution at a point, at a particular pH, the 50% of the molecules will be cationic form and 50% will be in zwitterionic form. The pH is known at that point is known as the pK1 with regards to the COOH. Okay, with regards to the COOH. In other words, because you are adding hydrochloric acid, which is a strong acid, is going to increase the acidic characteristics of the amino acid. So this pH is known as the pKa1. Now, if the titration is done at the isoelectric point with OH, remember that at OH point, at an isoelectric point, the net charges is zero. Okay. So when you do it, the same experiment with sodium hydroxide, it will be that at a point you will have 50% of the molecules will be in the anionic form, while 50% will be in the zwitterionic form. And at that point, you have the second pH change or buffering change, which is known as the pK2. And this pK2 will be in regards to the amino group, because you remember we said amino acids, they have two functional group. One is the carboxylate group and the other one is the amino group. So when you look at the structure which you're seeing right now, okay, now you can see at the base, you can see equivalent of hydrogen ion and the equivalent of hydroxyl ion, all going from a point zero. Okay. Now what we mean here is that if you begin to increase the pH, look at the right side of this plot. Okay, if you begin to increase the pH by adding, or rather by removing the hydrogen ion concentration, you're moving to the right, you're removing hydrogen ion concentration and you're increasing OH. Okay, in other words, you're moving from pH 0 probably to pH 14. Now, as you keep increasing this pH, you will notice that is by removing hydrogen ion. You will notice that there will be a point, a buffering range. The buffering range, which is at this point, where you are increasing the pH, you're increasing the OH amount, okay, but pH is not increasing. Are we following? If you're moving from here down to this place, you're reducing acidity and you're increasing alkalinity. Okay. Now, when you start doing that, you will get the point where the amino acid will begin to resist any change in pH, and this is our pH1. So after some time, you will notice that there will now start to be an increase also in pH, and you continue to add your OH, you will get the point that you will notice a jump. For instance, this is pH 4 and you notice a jump from pH 4 to pH 8. Okay. Now, the midpoint in this place you have point is where you have your isoelectric point. Isoelectric point. Remember, we say you can call it isoelectric pH. So at this point where there was sudden increase, you will still notice that increase will reduce. It will no longer be as high as it used to be. Okay. In other words, you notice that the amino acid begins to adjust itself again. It begins to adjust itself again to reduce the change in pH. Remember, The pH here is increasing because you're increasing OH, you are adding OH to it, and that is the second pH group, the second buffering zone, which is your pKa H. Okay, before you notice any other increase. So now for the monoamino monocarboxylic group, it has one amino group and you have one acidic group. So when you're adding OH to it, you will notice that the acid zone will be trying to adjust, to neutralize the effect of what you're adding. So in order to calculate the pi, the pi will actually be when you have, for we are looking at the monobasic amino acid, right? Okay, so the buffering action will be maximum around the pKa1, okay, and also at pKa2, which we have seen. But the pi, which is the isoelectric point or the isoelectric pH, will be the pKa1 plus pKa2 all divided by 2. That is for glycine. So if you're given the pKa1 and given the pKa2 of any monocarboxylic amino acid, you can be able to calculate the pi of such amino acid. So, but there are some of amino acid that has charged side chain. We will look at how you can calculate that. Okay, now, so for instance, if you have the pKa for the pKa for the amino, for the carboxylic group, and that of the amino group, it is, you can see that the carboxylic group is also within the acidic region, and the carboxylic group is within the basic region. Okay, now if you're given the, for those that has 3 because of the side chain, with this information you can always calculate your pi. So pi is just the area, sorry, is just the pki is just the average. For alanine, it will be this plus this divided by 2. Now, but for amino acids that have side chain, the shortest way to calculate the pi of such amino acids is to find the average of the amino acids that are close to each other. For instance, aspartic acid, the pi of aspartic acid will not be this plus this. No. Because the amino acid, the pKa of the carboxylic group is close to each other, you have to add this and this. While because 10 is far away from this and this. So if you are adjusting the both of them, it will already adjust them before 10, and you will still have the effect of that of the side chain. So to calculate that of the glutamic acid, you will see that 2.2 is closer to 4.3, so you have to add the 2 and find the average. But for something like cysteine, you find out that calculating the PA, you have the alpha group, the pKa as 1.8 in cysteine, 1.8, but you see the amino group is 10.8. And when you look at the side chain is 8.3. Okay, so it will be 10.8 plus 8.3 divided by 2. So you'll always look at the side chain, the one that has the side chain. If the side chain is tilting towards towards any of the values, whether acid or base, okay, you look at the value of the alpha group pKa, and then you will add it up. So for the ones we have here, it is only the 3.9, that is aspartic acid, and the glutamic acid that you are going to add with the alpha group pKa, okay, to find the average. Remember, it is not finding the average of the three you're going to look at. You're not going to find the average of the three for those that have it. It's always finding the average of two that are close to each other. Okay, sorry about that. Now, optical properties of amino acid. Now, the alpha amino acid, the alpha carbon of each amino acid is attached to 4 different groups, and like I said in the definition, and that's what makes it chiral or optical active of that carbon. So, but we have already established that glycine is an exceptional amino acid because it is not optically active, it is optically inactive. Now, amino acids with asymmetric center at alpha carbon can exist in 2 forms. You have the D form, you have the L form, which are mirror images of each other, and we call them the enantiomers. Okay, they are mirror image of each other, the L group and the D group. Now, all amino acids found in proteins are in the L conformation. That is why we say L-alanine, L-glycine, and so on and so forth. Not that we do not find the D form, but the D form is very rare. You can only find it in some cell walls of some bacterial cells. So we say the D amino acids are found in some antibiotics and in bacterial cell wall, but it is not general. In general, what you find is the L form. Okay, so now when we look at this, let's assume that you have a mirror here. You can see that the L form is the mirror image of D form. Okay, so but we are going to look at how we draw the structure very soon when we come to class. It will be more clearer then. Okay, now let's look at the reactions of amino acids. Because of the different chains that surround the different groups that surrounds the alpha carbon, you can actually see that amino acid has about 4 different types of reactions. Okay, now the first one will be reaction due to the amino group. We have reaction due to the carboxylic acid group. We have reaction due to the side chain, and also we have reaction due to both the amino group and the carboxylic group. Amino group of one amino acid reacting with the carboxylic group of another amino acid. So let's look at that. Now we have oxidative deamination. The alpha amino group is removed and a corresponding keto group is formed, and that is formed. So you have the alpha keto acid product or produced is either converted to glucose or to ketone bodies or is completely oxidized. Now, what do we mean? Remember, we classify some amino acids as ketogenic or glucogenic. Okay, so we are saying that in a reaction which is known as oxidative deamination, the amino group of amino acids could be removed and converted to keto acids. Okay, alpha keto acid. Now we equally have another reaction that involves the amino group. You have the transamination. We have the transamination, which is the transfer of amino group of one from one amino acid to the keto group of another keto acid. Okay, for instance, when you look at the diagram here, you find out that this amino acid is used to replace this keto group, and that is why the R1 amino acid here is now a keto group, while the keto acid is now the one that is having the amino group. So that is it's called transamination, where the amino group of one amino acid is transferred to the keto group of a second amino acid, so that keto group becomes an amino acid and the initial amino acid becomes a keto group. Now this reaction is always catalyzed by a transaminase. You will get this to know this in your third year, and you will do a lot of tests, okay, especially liver tests on this reaction. Now we are still looking at amino acids that are involving amino groups. Now, formation of carbamino compounds. Now CO2 binds to the amino groups on the globin chain of hemoglobin to form carbamino hemoglobin. Okay, and that is the way it is transferred, or it transfers carbon 4 oxide from the cells to the lungs. Now this reaction takes place at an alkaline pH and serves as a mechanism of transferring carbon dioxide from tissue to the lungs by the hemoglobin. So now when you look at this is representing the amino group in the hemoglobin, and that is where the carbon 4 oxide binds and is going to form this group. It's going to form the carbamino group. Okay, you can see the CO2 is attached to the amino group. Okay, now let's continue. What kind of rubbish is this? Okay, let's continue. Now the reaction involving the carboxylic group. You have one, you have the decarboxylation. When you remove the carboxylic acid group of an amino acid, you will equally have the process is known as decarboxylation. Okay, now amino acid undergoes alpha decarboxylation to form corresponding amines. Example, the glutamic acid, you can remove the carboxylic acid, and when that happens, you will have what is known as GABA, and GABA stands for gamma alpha butyric acid. Gamma alpha butyric acid. Histidine can also be carboxylated to form histamine, and this tyrosine can form tyramine. These products are very important in the body. Now, there are also the formation of amide linkages, and those formation of amide linkages, that non-alpha groups of amino acids can react with ammonia by condensation reaction to form the corresponding amines. For instance, aspartic acid can react with ammonia to form another amino acid that you know as asparagine. The same thing with glutamic acid. Remember that this glutamic acid and aspartic acid, they have dicarboxylic acid groups. Okay, so one of the groups can also react, can also be used to form the amide bond, and then form the corresponding amino acid. For instance, aspartic acid can form asparagine, while glutamic acid can form glutamine. Now, the assignment that you're going to be looking at in this place is to write out the reaction in which aspartic acid is converted to asparagine and glutamic acid is converted to glutamine. Also, write the equations in which glutamic acid is converted to GABA, histamic acid is converted to histamine, and tyrosine is converted to tyramine. Tyrosine is converted to tyramine. Okay, we also have the reaction due to the side chain. Remember that there are some amino acids that contains OH on the R or the side chain. Something like serine and threonine, they can form esters with phosphoric acid, in the formation of phosphoproteins. It is at that point where you have OH, that is where the reaction takes place. And most of the times, most enzyme inhibition takes place if the OH is in the active site of the enzyme. So the reaction can occur there, which will lead to the inhibition of such enzymes. So the OH group containing amino acid can form glucoside at the O, and then with the OH, thereby forming what is known as the O-glucosidic bond with carbohydrate residues. So you find out that this reaction we are talking about is occurring at the side chain of the amino acid, so they can form esters. Now, another reaction that can occur at the side chain will be the one that is due to the sulfur group or the SH group. Okay, now these ones that has SH or sulfuride group can form disulfide bonds. They can form disulfide bond. That is why cysteine, which is a sulfur-containing amino acid, can form a dimer, and that dimer is known as cysteine. Okay, so the dimer is just linking the sulfhydryl groups, two of them coming through and forming disulfide bond. So two cysteine residues can get connected to form two polymer chain, and then that is what we actually describe as cysteine. Okay, cysteine can then help to form cysteine. So you can look at the reaction, one cysteine, another cysteine, there will be an oxidation, and then the sulfur will join together. The sulfur will join together. Okay, look at it, this sulfur and this sulfur. When there is an oxidation can be removal of hydrogen, can be known as oxidation. So when it happens, we form a sulfur group or sulfur bridge. Okay, now another type of reaction that you see at the side chain is transmethylation. Methyl group in methionine can be transferred after activation to an acceptor for the formation of important biological function. Okay, so you can see that the side chain of methionine, of homocysteine can methionine of homocysteine, the sulfur group of homocysteine, which is part of the side chain, okay, can accept a methyl group from folic acid, and the enzyme that helps them to do that is known as methionine synthetase. Methionine synthase. Okay, this methionine synthase is what helps in the formation of the amino acid methionine. Okay, so it's another type of reaction, transmethylation, because it transforms the methyl group, from folic acid, okay, methylated folic acid, to homocysteine. And then the homocysteine is what is going to use to form the amino acid methionine, and then you have the tetrahydrofolate that is formed. Okay, very important reaction in the body. Now we still have the reaction due to the side chain, okay, where you form the amino acids of one amino group reacting to the carboxylic group of another, and that will lead to formation of a peptide bond. It will lead to the formation of a peptide bond. You can look at it to see this is an amino acid, okay, and this is another amino acid. Now the carboxylic group of this amino acid forms a bond with an amino group of the second amino acid, and in doing that, water is released. Okay, it is actually a dehydration reaction, and when that happens, something that is called is known as a peptide bond. So the peptide bond links one amino acid with the other amino acid groups. So this peptide bond that is formed is very important. That is the only way one amino acid can connect to another amino acid, with the formation of a peptide bond and the release of a water molecule, and that is a dehydration reaction. Okay, so we look at special functions of amino acids. Number one is that they are incorporated into tissue protein. Okay, now something like niacin serotonin, melatonin. These are components that are very important in the body, and then they are normally formed from some amino acids such as tryptophan. We have melanin, thyroid hormones, catecholamines are synthesized from tyrosines. Okay, we have the neurotransmitter, the one I talked about, the gamma, the the ones we have seen when amino acid groups is removed to form the amine group. So we have the GABA, which is a neurotransmitter, is synthesized from glutamic acid. Now also we have something like nitric oxide, which involves in smooth muscle relaxation. Okay, now it is formed from arginine, and that is why most people take fruits that is rich in arginine. Something like what do we call this? Cucumber is rich in arginine. Okay, and then the smooth muscle relaxation, when the smooth muscle around the pineal gets relaxed, blood flows into the pineal, giving it a very strong erection, and that is why so many people indulge in eating a lot of cucumber during sexual activity. Now they also act as precursors for heme, creatinine, and glutathione. Okay, glutathione is made up of three amino acids. Now you have the porphyrins, you have the purines, you have the pyrimidines. All these things are being formed from amino acids. Now, in carrying out the test of amino acids, which I know you will do in the lab, you're going to talk about the Neihydrin reactions, okay? And this gives an alpha amino acid. It gives a reaction with all alpha amino acids. That's why that is a very important reaction that we actually use in the testing of proteins. You have the xanthoprotic test, you have the million test. Million test will also confirm that the amino acid we have contains or that the protein you have contains tyrosine. Okay, now you have the Burette test. Okay, you use them to determine amino acids that are not free. That's the ones that are bound to protein. So million test and the Burette test is normally used as protein test. You have the Sakaguchi test, which is used to determine protein, but what it attacks or what it confirms is the presence of arginine. And then you have the Hopkins Kohler reaction that confirms the presence of tryptophan. All these are all protein tests, but there are particular amino acids that they target in that protein. Now we'll have lead acetate test, which is actually targeting the sulfur containing. It targets cysteine and cysteine, but not methionine. Okay, now we have the nitroprusic reaction, which actually looks for the sulfur in the amino acids. They are all color reactions. Sorry,